I'm Mark Laing. I'm the Professor of Plant Pathology at the University of KwaZulu-Natal in Pietermaritzburg in the Agricultural Faculty there. My specialization is in biological control of a whole range of pests and diseases. And one of the projects we're working on is the biological control of mastitis, which is caused by Staphyloccocus aureus largely, but a number of other bacteria that affect cows and humans. The big problem in the world is that the disease has been controlled for the last 30, 40 years using antibiotics. And unfortunately, there's a development of antibiotic resistance across the world, including South Africa. So we need to come up with an alternative that gives us control of mastitis. And the only really good long-term option available to us is biological control, which is using natural organisms to control a problem. And in this case, we're going to use bacteriophages, which are specialist viruses that land on, attack, and kill target bacteria. They're very specific. So a phage that kills Staph aureus will not kill Streptococcus, but that's a big advantage because it doesn't cause detrimental activity to other organisms.
I'm Iona Basdew. I'm a postdoctoral researcher in the Discipline of Plant Pathology at the University of KwaZulu-Natal. My major research focus for the postdoc is the biological control of mastitis using bacteriophages. A phage resembles a lunar lander. Imagine a little rocket landing on a planet. It has its little legs. It lands onto the surface of the bacterial cell via attraction of sugars on the bacterial cell. And upon landing on this specific bacterial cell, it injects its DNA into the host bacterium, and this DNA then initiates a replication cycle which breaks down the cell components of the bacterium itself to regenerate new bacteriophage particles. This whole process from landing onto the bacterial cell to regeneration of new viruses takes approximately 20 minutes. So in a matter of 2 hours, you could have a 1,000-fold increase in the number of phages that are released from a particular bacterial cell. So once these bacteriophages have replicated within the bacterial cell, this increased pressure within the bacterial cell causes the cell to rupture, and in so doing, new bacteriophages are released into the surrounding environment. And each new bacteriophage is able to bring about a new replicative cycle where it itself produces up to 1,000 progeny, and in so doing, perpetuates the cycle and perpetuates the control as well. The current method that is most commonly applied to measure the level of mastitis in a dairy cow is the use of somatic cell counts, which is a measure of the number of white blood cells present in the milk at milking. Now, we have discovered through our studies that the somatic cell count is actually a delayed indication of infection that had happened prior to the actual sample being collected. We actually discovered that cows that had exhibited a high somatic cell count, in up to 60% of the cases, actually had zero bacterial growth, zero staph, zero strep, just basically zero bacterial growth. And that led us to believe that perhaps the somatic cell count is an inaccurate measure of the actual state of disease of the cow in real time.
Bacteriophages also have a problem with resistance because the bacteria change the sugars on their coat, which makes them undetectable to their normal bacteriophages, with the result that we needed a strategy to manage the resistance. The way we do it is to have a cocktail of phages to start with, and then constantly rotate the members of this cocktail, because there are thousands and thousands of strains of bacteriophages out there. We have to isolate the most effective ones that can tackle different strains of Staph aureus.
My name is Ndlela. I'm a master's student in the Department of Plant Pathology, School of Agricultural Earth and Environmental Sciences. I am involved in the mastitis project, where my role is to research about the use of bacteriophages and bacteriocins as alternative control to control bovine mastitis in dairy herds.
My name is Mduduzi Shinga. The primary aim of my project is we're trying to look at alternative methods, diagnostic method that will detect mastitis in cows.
I'm Caryn Shacklock. I'm the vet in the milk lab at Allerton Veterinary Laboratory. Allerton is a government-owned veterinary laboratory focused on animal disease diagnostics and surveillance. There are 13 sections to laboratory. The milk lab is just one of them.
If you've got high clinical incidence of clinical mastitis and you're not picking that mastitis up, you're not treating that mastitis, that can hurt you financially, and that can be up to 4 cents a litre, which at the end of the day, if you're sending 1 million litres of milk a year, that's a lot of money that you're wasting. So you have to have systems in place for identifying mastitis. You have to know what pathogens you're dealing with, whether they're environmental or just the other pathogens. That's why working with UKZN is really important to us, so we can get a handle on the pathogens that we're dealing with and we can treat them correctly.
Biological control is very often stops at the gates of the university and doesn't get out to the farmer. In our case, we're working with an industry partner who has a full-time professional virologist who specialized in phages and human phages at that. And they're working side by side with us to work out how to commercialize bacteriophages to get them to the farmer. So our goal in this research is not just to find potential therapies, but to actually develop commercial products which can be commercialized and released to farmers.